I was kind of talking about the last time, we're going to take a little bit of look at the internal structure of cells. Again, not something that we're going to be differentiating too much when it comes to actually having to identify this stuff. I mean, again, nucleus, cytoplasm, some of the things like that, yes, definitely would be fair game. But we're not going to be looking at micrographs of all this stuff and trying to identify intermediate filaments versus the plasma membrane versus smooth endoplasmic reticulum, something like that. Main thing is, a lot of times there's a lot going on inside these cells that, again, depending on the tissue type we're looking at, you see certain structures that are going to highlight one of these internal cellular structures. Again, Nissl bodies being kind of one of the better examples in nerve cells. So when we start talking about cell, to me, really the most important thing here is going to be the plasma membrane. It's what defines the cell as a cell. This is the outer covering of the cell. It is a phospholipid bilayer, so you have a phosphate head, a tail, phosphate head, and a tail. You put those two things together, that's that bilayer. Plant and animal cells both have this structure. Plant cells would have a cell wall surrounding the outside of that as well, made of cellulose. And again, a lot of the membrane structures on the inside of the cell are going to be a similar thing here. So again, phospholipid bilayer, they talk about this being a fluid mosaic model, so you can see the phospholipids right here. These are free to float around in there. There is proteins that are either integral going all the way through or peripheral going part way through of this membrane. But again, each of these phospholipids heads is free to move around here. These things float around in there, so it is getting, to me, a lot like if you were to blow a bubble where everything's swirling and moving around in there. That's really what's going on with these phospholipids when you're talking about the plasma membrane. Animal cells will have cholesterol in those to help kind of hold it together a little bit more. Again, this is all stuff we're not going to see when we're looking at under the scope, but you will be able to define where the plasma membrane is, where one cell begins and one cell ends. So again, this is what makes a cell a cell. If you were to rupture the plasma membrane, there is no homeostasis. The inside is the outside, and that, in almost every instance, kills whatever this cell is. So they give us a kind of a rough look under a light microscope. You can see where one cell ends and one begins. We can't see the actual microstructure of that with the phospholipid bilayer without an electron microscope, but we can see where one cell begins and one cell ends. Again, cell membrane, you can see it on these ones here. These are probably some kidney tubules. Again, you can see here's the cell membrane on each of these ones here. A lot of times lights up a little bit as you see a little bit darker with the stain. One of the other things we can see really well in these ones is the nucleus also. So the nucleus is where that genetic material is. A lot of times this is going to take on that basophilic stain, this dark stain, that purple stain. Usually be pretty well visualized on a lot of these slides here. Sometimes you will have this DNA condensed a little bit more if the cell's getting ready to undergo mitosis, and you can see the actual chromosomes. But more often than not, you're going to see this kind of speckled kind of darker and lighter area nucleus with that chromatin, the euchromatin and the heterochromatin in it. That's going to give you those different kind of dirty, kind of dishwater looking nuclei. Again, when it gets really well condensed, you can see the actual chromosomes with the cell undergoing mitosis. We'll talk about that a little bit in the second half, second show of this. But in normal instances, it's going to be subdivided into heterochromatin and euchromatin. Heterochromatin is the denser, more darkly staining. So on the electron micrograph over, you can see heterochromatin is the dark areas, euchromatin is here. A lot of times we'll see these on certain cells as well if we're zoomed in a little bit more. The other thing we'll see a lot of times in this nucleus, and you can see it pointed out on both the micrographs above here, is the nucleolus. This is a darker staining region of the nucleus that is involved in making ribosomal RNA, which is going to be important for the ribosomes, and then filling in this membrane where around this chromatin and everything else is what we refer to as the nucleoplasm. So that rest of the nucleus that is not DNA, not the nucleolus. So you can see the nucleus on this one right here with the nuclear envelope. The nucleolus is the darker staining there, and you can see the heterochromatin and euchromatin, the light and dark areas on this one. And again, you can see it on the electron micrograph as well, the heterochromatin, the euchromatin, and then again, the nucleolus would be the darker staining region right there. Again, letting you kind of see the overall what's going on here. Again, I'm not planning on doing a quiz where we look at this stuff so much. We'll have a lot of slides to look at on the first test that will be of the actual tissue types. So this is, again, is just a review of cellular structure to kind of get everybody on the same page. So surrounded by the nuclear envelope, the nucleus is. This is kind of what's deciding what can get into the nucleus, what cannot. It is a very selective membrane. It has pores or openings within it called the nuclear pores. I always kind of say the nucleus is kind of like a CEO. His or her office is kind of a nice corner office, has a administrative assistant that can decide who can get into the office, who can't through that office door. That's what kind of like the nuclear envelope, and the pores within it are kind of serving to do, really deciding what can get in there. We want, because of all the DNA being in there, we want the nucleus to be a rather selective place where we can't let every type of chemical or component getting in there. What kind of gives the cells their structure is going to be a lot of these different filaments. We are going to have what they kind of, you can see microtubules, intermediate filaments, as well as microfilaments. Going to be putting this all together. A lot of these protein filaments are going to make up this skeleton or structure of the cell, this scaffold that's going to help it hold in place. Very rarely do we see these. Generally with light microscopes you can't see them particularly well. Sometimes in cells where they're very, very obvious, you can see them a little bit more. Again, sometimes we talk about actin being a microfilament. That is one of the ones we can visualize in muscle. These myosin and actin filaments are types of filaments in the muscle cells that are a little bit more obvious, involved in kind of pulling in or out on these ones. But those would be part of one of those thin filaments. Microtubules are another level on these ones here. These are hollow structures, help make up part of the cilia and flagella that you would see in certain cells, as well as the centrosomes involved in mitosis. Intermediate filaments kind of in the middle here, in between these microtubules and the thin filaments. Again, provide a pathway to help move some of the cytoplasm. And the other thing we'll see is a lot of times we use different types of ATP-driven cellular motors to move things, like in a nerve cell, using the cytoskeleton as a kind of a, if you want to think about it as like a minecart track to move things around within the cell. We can sometimes use that with cellular motors and vesicles, those little circular, spherical, membrane-bound structures inside of a cell that you can actually pull along. A lot of times in certain cells we will see the ribosomes, not the specific ribosomes, but where they're at. So you can see the Nissl bodies on this nerve cell here. These are darkened areas of rough endoplasmic reticulum, which is has embedded ribosomes in the wall. We talk about some of these being involved with the endoplasmic reticulum. That is referred to as the rough endoplasmic reticulum. Outside of that, we see a lot of free ribosomes just inside the cellular fluid, that cytoplasm. The endoplasmic reticulum is this network of tubules divided into two different parts. You have the smooth endoplasmic reticulum, which is involved in kind of lipid and carbohydrate synthesis. In certain cells, it's calcium storage, where the rough endoplasmic reticulum has ribosomes embedded in its wall. This is involved in making proteins that are going to go into vesicles, or extracellular proteins that are going to be sent outside the cell. These proteins are made into the inside of this rough endoplasmic reticulum. We can then package them and move them around the cell, sometimes to, for example, the Golgi apparatus. So you can see here, a lot of times in these cells, the more rough endoplasmic reticulum, the darker it's going to stain. So cells that don't have a lot of this tend to stain a lot more with that eosin. As we get more and more of it, you will get a little bit more of the hematoxylin stain in the cytoplasm, making the cell look a little bit darker. And again, this is showing you an electron micrograph with these rough endoplasmic reticulum. You can see the ribosomes embedded in the membrane. Again, that's what makes it different from the smooth endoplasmic reticulum, which is probably up here, where you do not see any of these little bumps along the membrane. And again, it's pointing some of this out here. Sometimes it's a lack of staining that you would notice because there's no ribosomes there. And if it's mainly lipid, it doesn't stain particularly well. Again, not going to make you try to differentiate that or find that on a micrograph here. But you can see the smooth endoplasmic reticulum doesn't have the ribosomes in the wall, but otherwise looks a lot like the rough endoplasmic reticulum. Sometimes we are going to send a vesicle of something to what is called the Golgi apparatus. This is a kind of, to me, it's like a little stack of pancakes. It's these membranous sacs that are stacked on top of one another. Again, when you see the electron micrograph of it, it's really easy to see that. These a lot of times are involved in repackaging and moving substances to different areas of the cell. Like it says here, sometimes the pale region near the nucleus is where we can see this. Again, I don't expect you to be able to differentiate this. This pale region close to the nucleus is where the Golgi is at. Again, it's made of lipid. That's part of the reason it's not going to stain particularly well. It's kind of pointing it out on this one. But what we're really looking at is you have this stack of membranes. To me, it looks like a pile of silver dollar pancakes that got dropped. What's happening is vesicles are coming in one side, being repackaged as they move through these layers here, and then getting targeted to, let's say, the cell membrane or a lysosome or something else out this other side. So that kind of gives you an idea of some of the basics here. What we'll do on the second half of this is we'll work through a little bit more of these cellular structures and talk a little bit about mitosis. Again, this is just, again, trying to get everybody back on the same page. If you've had AMP, if you haven't, depending on which bio courses you've had, so everybody's on the same page in terms of cellular structures. So as we start going to that next level of the kind of hierarchy here of tissues, that we're all working from the same base of knowledge on that one. So I will talk to you again next time.